Mr. Majewski's Anatomy 32 class lecture, Chapter 10, Part 2, Sarcomere, Types of Fibers and Disorders. So here again is a close-up of a muscle fiber, and we see within the muscle fiber many rod-like structures referred to as myofibrils that run the length of the muscle fiber. And it is these myofibrils that shorten during a contraction. If we take a closer look at a myofibril, we see that it is made up of structures called sarcomeres that are connected end to end to each other, forming a long chain of hundreds or even thousands of sarcomeres. And it is these individual sarcomeres that actually shorten during a contraction. Within a sarcomere, you see that it is composed of thick filaments and thin filaments, the thick filaments being thicker than the thin filaments. You also see some other structures found in the sarcomere. At either end of the sarcomere is what is referred to as the Z-disc. So two sarcomeres meet up at the Z-disc. And then in the middle of the sarcomere is a structure referred to as the M-line. From this we can see a number of other interesting features of the sarcomere. For instance, there is what's called the A-band. The A-band is the full length of the thick filament, while the H-zone is where you only have thick filament present and no thin filament. And that is found around the M-line. You also have what is referred to as the I-band. This is where you only have thin filament present, and this is found around the Z-disc. So if you look at an electron microscope view of a sarcomere, you do indeed see the Z-disc structures at either end of the sarcomere, the M-line structure in the middle, and you can tell that the A-band is present and is the full length of the thick filament, the H-zone where you only have thick filament and no thin filament, and the I-bands where you only have thin filament and no thick filament. And it's pretty clear that there's a slight color difference between these various areas of the sarcomere, and it is these differences that make up the striations that you see in skeletal muscle cells and cardiac muscle cells. Now, if we look at what makes the thick filament, we see that it is composed of many proteins called myosin proteins. And these myosin proteins are one of the two contractile proteins that are involved in causing the sarcomere to get shorter. And so any one thick filament will have hundreds, if not more, of myosins forming it. The thin filament is composed of actually three different proteins. It is composed of actin, which in this case is seen as these yellow ball-like structures. Actin is the second contractile protein, and it has what's referred to as myosin binding sites, so that the head of the myosin protein can actually bind to the actin protein. You also have two regulatory proteins found in the thin filament. You have the tropomyosin, which in this case is shown as a long brown rod, and the tropomyosin actually covers the myosin binding sites, so it prevents myosin from binding to actin. The second regulatory protein is troponin, here seen as sort of a blue t-shirt-like structure. And what troponin does is when it is bound to calcium ions, it'll change its shape. And when the troponin changes its shape, it'll force the tropomyosin to shift position so that it no longer blocks the myosin binding sites, thereby allowing myosin to bind to actin. And this will then allow for the contraction cycle to occur. So here we have a state where myosin in the thick filament is bound to ATP, the energy currency of the cell. And the thin filament, the calcium ions are bound to troponin, thereby causing it to force tropomyosin to shift out of position. So now the myosin binding sites on the actin are open and ready to be bound to. Step 1, the myosin head will hydrolyze the ATP, thereby becoming energized. It'll be taking the energy from the ATP. In this process, the ATP will be broken down into ADP and inorganic phosphate. This causes the myosin to change its structural shape so that it is now ready to bind to the actin. In the next step, the myosin heads will bind to the actin in the thin filament. This forms a cross bridge. You now have a bridge between the thin filament and the thick filament. When this happens, the inorganic phosphate will be released from the myosin. In the third step, called the power stroke, the myosin will now change its shape again, moving the thin filaments toward the center or the M line of the sarcomere. And in the process, the myosin will release the ADP. And then finally, ATP will bind to the myosin head, thereby causing it to release the actin, restarting the whole process. This contraction cycle will continue for as long as two things are at high enough levels. The ATP, to allow the release from the thin filament, and the calcium ion, to allow the troponin to keep the tropomyosin out of the way of this interaction. This is referred to as the sliding filament mechanism, or sometimes also known as the sliding filament theory, where you start off with a relaxed sarcomere. The calcium gets released, binds to the troponin, causes the tropomyosin to move out of the way, and now we start getting a contraction going. So we get those cross bridges formed, and as you can see, with a partially contracted muscle, the H zone and the I band are starting to get shorter as the overlap between the thick and thin filaments increase. And then with a fully contracted muscle, you see that there's very little to no H zone, very little I band, and the thin filament and thick filament are almost completely overlapped. Key thing to point out is that the filaments remain the same length. The filaments do not change at all in terms of length. It is only the sarcomere that changes in length. It is the sarcomere structure and only that structure that gets shorter. Now, eventually the contraction cycle must end. This happens when you no longer get the nerve impulse that leads to the generation of the muscle action potential. Without this muscle action potential flowing through the sarcolemma and into the T-tubules, you no longer signal the release of calcium ions from the sarcoplasmic reticulum. When this release stops, calcium will only be entering the sarcoplasmic reticulum. So basically, the sarcoplasmic reticulum is always bringing calcium ions into itself, but not at as great a rate as it can potentially release the calcium. So when it's no longer releasing, it'll then remove all the calcium ions from the sarcoplasm, ending the contraction. Rigor mortis. Well, it turns out rigor mortis occurs because after you die, your cells are no longer maintaining their preferred states, and cellular membranes are kind of leaky. So the calcium ions escape the sarcoplasmic reticulum, triggering the contraction of the muscles. However, once your cells are dead, they no longer are producing ATP. And so without the presence of ATP, you can't have a release of those cross bridges, so the muscles end up stuck in this contracted state. Eventually, though, the cells will degrade and enzymes will break down the sarcomere itself, and once that's broken down, the muscles will then relax again. Muscle contraction types. Well, there are two big categories. Isotonic contraction being one of those, where you have a tension that is constant as the muscle changes in length. So in this case, the bicep getting shorter as you lift the dumbbell. Now, the isotonic contraction can be itself broken up into two types. The concentric isotonic contraction, where the tension is generated and is enough to move the object, and in the process the muscle shortens. There's also the eccentric isotonic contraction. Here again, you have enough tension being generated, but this time the length of the muscle increases, but slowly. So this is the controlled lowering of an object. The second big category of muscle contractions is the isometric contraction. This is where the muscle is generating a lot of force, a lot of tension, but it is unable to change in length. It can't overcome whatever it is pushing against, such as that wall. There are also three types of skeletal muscle fibers. The first type is what's called slow oxidative fibers, or type 1 fibers. They are red in color because these cells are high in myoglobin. They have a lot of ATP, and they also get a lot of capillaries flowing to them. And they can contract continuously because they have all these energy resources, but they generate very little power. These are often used in activities such as maintaining your posture and aerobics. Then there are the fast glycolytic fibers, or type 2b. These fibers are low in myoglobin, have very little mitochondria. However, they do have a much larger diameter. So what they're able to do is produce a lot of power, but only for a very short time. And this is often used in weightlifting. And then there's the intermediate type of muscle fiber, called the fast oxidative glycolytic fibers, or type 2a. And they're basically an intermediate between the two, and even have sort of a pinkish color. And these are more for sort of intermediate activities, like sprinting or walking. So why do the slow oxidative fibers, the type 1, possess such high levels of myoglobin and mitochondria? Well, it turns out, as we said before, the myoglobin binds oxygen, and the oxygen is needed by the mitochondria to produce the ATP, or the energy currency of the cell needed to get our contractions. When you exercise, you can change the characteristics of your muscle fibers. So for instance, if you focus on endurance exercises like running or swimming, you can slowly convert some of your fast glycolytic fibers, or white ones, into the fast oxidative glycolytic fibers, or pink ones, which then will help improve your ability to do those endurance activities. On the other hand, if you're doing rapid strength exercises like weightlifting, what happens is your fast glycolytic fibers will actually increase in diameter, which is called hypertrophy. And that's why bodybuilders have such large muscles. Cardiac muscle fibers. So, as we know, they are different from skeletal muscle fibers because they're branched, they are controlled involuntarily, they display autorhythmicity, they can generate their own muscle action potential. And while they possess, as an organ, the endomysium connective tissue and the perimysium connective tissue, they lack that final outer epimysium connective tissue. And of course, they have those intercalated discs. And these are important because at the intercalated discs are gap junctions. And this allows that muscle action potential to flow between the connected cells in the heart tissue. Smooth muscle fibers. Smooth muscle fibers lack the striations that we expect. They are long tapered cells with only one nucleus, and again, we have no control over them. They also lack the sarcoplasmic reticulum and T-tubules. So how do they work? Well, they do have thick and thin filaments, and these thick and thin filaments are attached to intermediate filaments, which form part of their cytoskeleton. And the intermediate filaments are attached to dense bodies found in the sarcolemma, or the plasma membrane of the cell. And so when these cells are signaled to contract, the shifting of the thick and thin filaments over each other then pull on the intermediate filaments. The intermediate filaments then pull on the dense bodies, and the dense bodies end up pulling the cell together, forming a shorter cell. But this whole process is much slower than you'd find in skeletal muscle cells. There are two types of smooth muscle fibers. You have the visceral smooth muscle tissue, which have the gap junctions present, so the muscle action potential can flow between cells. They therefore only need one neuron to signal the entire tissue structure, versus the multi-unit smooth muscle tissue. Here there are no gap junctions, so each individual muscle fiber needs its own axon interacting with it. Aging. Well, as you get older, you lose muscle mass. And as this muscle mass is lost, it is often replaced with fibrous connective tissue and fat, so that you don't notice this loss. However, fortunately, it can be reversed or delayed with exercise. Finally, there are some muscle disorders. There's muscle cramps, which are a hyperexcitability of the muscle end plate. There's overuse, which is tearing the muscles. There's muscular dystrophy, which is a disorder that leads to muscle replaced by connective tissue. And there's myofascial pain syndrome, which is inflammation that leads to much pain. We're done.